Good afternoon, everyone. My name is Stephen Liang. I'm the Director of Radiochemistry, Nuclear Medicine, MGH. I'm also the Associate Professor of Radiology, Harvard Medical School. It is my great honor today to present my work in this symposium, which is dedicated to Dr. Saul Hertz, the founding director of MGH Thyroid Unit, for his pioneering work for radioiodine therapy. I want to show you a flow chart from bench to bedside discovery these days. Now, we already discovered the molecule from molecular or cellular level, and then we test our hypothesis in disease models in preclinical setting. And then once we prove its efficacy and the safety, we translate to clinical trials. Now, in related to radioactive halogens, we use F18, iodine-123 for imaging work, and then we use iodine-131, and even for alpha-based therapy, astatine-211 for radiotherapy work. And the development chart I show you here is not new for these days, but let's imagine back to 80 years ago. This is truly pioneering and instrumental, especially during 1940s, during World War II. When Dr. Hertz developed the radiotherapy and also overcame all the barrier to translate it into human use. And in this short talk, I'm not going to review or reiterate Dr. Hertz's professional training and the practice, because you may find them in many publications or archives. Rather, I'd like to share some historical photos to highlight or recapture this important moment. I think the first figure you probably see in the other talks is the collaboration between MIT scientist Arthur Roberts and Dr. Saul Hertz on the right. So when they performed the preclinical imaging study or preclinical study in rabbit model. And on the right is that Dr. Hertz actually invent or successfully commercialize an instrument called multiscalar to testing the thyroid uptake. And also this the early demonstration how important a biomarker can be used for precision medicine or precision therapy, radiotherapy. And then this is from a 1942 newspaper. We can see a patient before and after the therapy. And also another photo to demonstrate the use. Now this is for President Bush, the thyroid treatment. Radio halides or halides are widely used in nuclear medicine and molecular imaging. And from the chemist's perspective, this is a very atypical periodical table. But from the radiochemistry perspective, we see this differently. As showing on the right, we see this, all the radioisotopes in different half-life, different energy levels. And I would say, depends on the nuclide, the halogens can be used for PET imaging, the positron emission tomography imaging, or single photon imaging SPECT, and also or radiotherapy like Dr. Saul Hertz used for iodine-131. And I would say the most frequently used radiohalide nuclides are fluorine-18, iodine-122 and 123, and iodine-131 for radiotherapy. Now, since we start to introduce the concept of this PET in the positron emission tomography, I'd like to introduce a little bit background on this. Now, it's an imaging technology developed by 1950s. They utilize a target molecule incorporate with a positron emission nuclide. And then we inject to in vivo target, for example, a human test subject or animal models. So we have this in vivo image of for the disease diagnosis or for the treatment evaluation. So we call those three elements will be the minimal list for the PET imaging. I would like to show you a workflow for PET imaging. Well, cyclotron is a particle accelerator which generate different isotopes. As shown here, we use medical cyclotron generate carbon-11, F18, or there's other technologies including generator-based or cyclotron-based, we can generate radiometals. Once we have those elements or nuclei. The radiochemist's job is putting those elements incorporate into molecules. Could be a small molecule, as shown in this slides, or large molecule, biological molecules. I mean, we can use chelator to chelate the radiometals. Once we have the tracer, or once we have the chemicals ready, we call it a labeled compound for most of the time for animal use, or it is for human use. We call it radiopharmaceuticals, radioactive drugs. And then the patient or the subject under the PET imaging scan. So what was happening during that, we call PET physics, is during the event, the positron annihilate with the electron. So it gives out two gamma rays. Now for this example, F18 decayed to oxygen 18 to give it one positron. So through a sophisticated reconstruction, we all know three specifics: a specific time, specific location, and specific concentration of our radio tracer. So through those information, we can reconstruct a 3D image, as I show you in this figure on the bottom right. We can see the whole body, or we can see the organ of interest as the brain image showing on this slide as well. Now, PET imaging, as in 2004, is recognized by FDA, one of the critical path for drug discovery. Now, from the PET imaging, we can use this invaluable imaging biomarker for early diagnosis, for cancer treatment, design, or for Alzheimer's disease, for example, neurodegenerative disease. And for drug discovery, we can really answer the very fundamental questions for pharmacokinetics and pharmacodynamics. Through the different stage of drug discovery, the PET imaging can serve a different function, as I'm showing on the bottom of this slide. I want to give you a very quick snapshot. If we label the F18 at two positions of the glucose, we call this FDG, F18 FDG. So this tracer is most widely used positron emission tracer in clinical. We can use this for oncology indication. We can use it for CNS indication, as showing on the F-dopa, as showing on the bottom. We can monitor. We can see the subjects different functions. For example, looking, listening, thinking, remembering, and working. Or when we incorporate F18 on different type of molecules, for example, dopa become F-dopa, or there's an amyloid imaging agent for other indications for Parkinson's disease or for Alzheimer's disease. And one of the ongoing challenging is to make F18 molecule on non-activated aromatic rings. Now, since this is for the talk for the ACS, American Chemical Society, I'd like to spend a little bit more time about the chemistry. For example, like the traditional way to put F18 on the aromatic ring is through electrophilic reactions. But those type of reaction is selectivity and specificity is usually very low. And the traditional way we use it for introduce F18 in a nucleophilic way is aromatic substitution. However, a demand for electron withdrawing group. Therefore, there's a continued need or urgent need to introduce F18 on non-activated fluorine items, which is very popular in many of the pharmaceuticals, not only in radiopharmaceuticals, even in other pharmaceuticals for traditional medicine. And this, I'd like to show this technology we developed at MGH. And I show you in the figure in the middle, we incorporate, we call auxiliary molecule to activate the iodine, we call hypervalent iodine to introduce molecule. And on the background, there's the molecule we're able to achieve in this technology. As a recap on my talk or my dedication, now I would like to quoting two remarks from my coworkers in the field. Definitely Dr. Hertz, the contribution has been inspirational for leading scientists towards cancer therapy. Also, due in no small part to Dr. Hertz's contribution, we have made significance to in the field for nuclear medicine. And at the end, I would like to acknowledge Hertz's family, two colleagues from Harvard, Dr. Anna Swartzmack, the past president of Northeastern section of the ACS, Dr. Alan Packer, the past president of SNMI. Also, I would like to thank the ACS, our Northeastern region, also the MGH Radiology, Nuclear Medicine, and also MGH Media Office. At the end, I would like to thank you very much for your attention. Thank you.